So last time we talked about cloning plants, and I want to reinforce a few things here because it's quite complicated and it does come up on the triple paper quite often. So let's imagine that this is an orchid, and orchids are commercially very, very valuable and they're very difficult to grow. So what I'm going to do is I'm going to take a piece of tissue from the orchid, their adult orchid cells, diploid cells, and I'm going to place them here into this Petri dish with an agar growth medium, and that agar growth medium has got within it plant hormones, auxins, which are going to encourage the tissue here to undergo cell division. So I'm going to get a mass of cells which are all genetically identical to each other and also to this original orchid plant. What I'm then going to do is transfer it to a second Petri dish, and the second Petri dish also has plant hormones, but they're different types, and this time they're going to encourage the growth of the root and the shoot. And what I then start to develop are these tiny plantlets. And each one of these tiny plantlets is genetically identical to one another and genetically identical to this original orchid plant. What I can then do is take those out of the Petri dish, transfer them into a soil medium. That soil medium is going to be rich in nitrates, phosphates, all the things that plants need, the minerals that they need for growth. And what I'm going to get then are a lot of these plants being produced from this original plant, and I can do this in a relatively short space of time. I've got to do it in very sterile conditions, but there are plenty of advantages of me doing this, which I'll come on to in just a moment. If you haven't already, please have a look at the Google Classroom and find this video, play this video, and that will reinforce this work even further. And what he does is he reminds us that we know about natural cloning within plants. We talked when we did plant reproduction about strawberry plants and runners and how they produce new strawberry plants which are genetically the same as the original one parent. We talked about potatoes and the tubers that come from potatoes and bulbs and corms. All of these are natural clones which happen within nature. Micropropagation is man manipulating deliberately using plant hormones, this process, to produce large numbers in a very short space of time. So the advantages: large numbers, short place of time. All of them have got the desirable characteristics. All of them are going to be free of disease. We can do this with endangered species so that they're prevented from becoming extinct. We don't need pollinators and we don't need a particular time of the year. So for example, Valentine's Day in February is not the normal growing time for roses, but we can manipulate cloning techniques in order to produce vast quantities of roses in order to meet the demand for Valentine's Day. We can also do cloning techniques with genetically modified plants. We'll talk more about genetic modification in a subsequent lesson, but that's also there as one of the things for us to talk about before we finish the syllabus. Disadvantages. We talked about disadvantages of clones previously. If you remember, we talked about the Irish potato famine, and that was caused by the fact that within Ireland at the time, all the potato plants were exactly the same as one another, genetically identical to one another. A new pest species called phytophthorans arrived, and that phytophthorans obliterated the entire crop and caused mass starvation. Now, what we've got in terms of microcloning is the similar idea. Because we've got genetically identical organisms, they're all susceptible to the same disease and to the same pest. It's also a technique that requires extremely sterile lab conditions, which are very expensive. And if you get any contamination, then the whole crop will fail. Now, moving on, the next thing for us to consider is cloning in mammals. And cloning in mammals is going to be illustrated using the story of Dolly the sheep. And the first thing to notice is we've got 3 sheep here, A, B, and C. And these 3 individual sheep are individual. They are genetically different from one another. And the purpose of this first female, A, is we're going to use her udder. We're going to remove a cell from her udder, and this is an adult diploid cell. And this adult diploid cell contains all the DNA that is required. Some genes might be switched on or switched off, but it's got all of the diploid genes that are required. We're going to take that out so we can extract the DNA. Meanwhile, female B, what her purpose is to produce eggs. So we're going to take from female B an egg cell. Remember, egg cells are haploid. They contain half of the chromosomes. But in any case, what we're going to do here is we're going to destroy the chromosomes inside. We're going to produce an egg which has got the nucleus removed. That's this one here. That's called an enucleated egg. And this egg, like all eggs, have got very special characteristics. The cytoplasm is rich in nutrition, and the cytoplasm contains many, many mitochondria. The cytoplasm also can be stimulated to develop by mitosis and produce new individuals. So what we need to appreciate here is that the biology of the cytoplasm of an egg cell is fairly special. We're going to take the adult full host of chromosomes and we're going to place it into this egg to produce this one here. So what we've got is an egg cell in a diploid condition, which is usually naturally what we're talking about here is the process of fertilization producing a zygote. But instead, we've got the egg with adult DNA placed inside it. But this is a diploid cell, which is then undergoing electric shock treatment, which causes that to divide by mitosis to produce a ball of cells, which will develop into the blastocyst. Now, this blastocyst is then inserted into female C. Now, female C has been primed with hormones, so her uterus is ready to receive this blastocyst. The blastocyst develops into the embryo. The embryo then develops into this lamb, which is here, lamb A. Now, the reason that this one is called A is because the lamb is genetically identical to this female here. So what we've done is produce a lamb using this this technique of cloning. Here we've got the four, you can read this for yourself, you've got the 4 stages that take place, and here is a video which again I suggest you go away and watch to reinforce everything that I just said, and I'm sure you'll have questions which you then come back to me with to answer. The last syllabus statement is real cutting edge stuff. So what we're talking about here is the use of cloned transgenic animals. Now, first of all, a transgenic animal is an animal which contains genes from more than one species. And I would urge you to go into this New Scientist article, and this unpacks the story I'm just about to talk to you about. We get a cow, and the cow has inserted into it a gene which ordinarily is expressed in the white blood cell. Now let's go back and think about white blood cells. White blood cells in our body allow us to defend our body against disease by producing antibodies. So our lymphocytes have got DNA within them that are expressed by, they undergo transcription and translation to produce antibodies. And antibodies are giving our body protection against microbes. The antibodies will attach to the antigen on the surface of the microbe and cause that microbe to be destroyed. So what we're doing here is we're taking a cow, we're inserting a gene, and that's a human gene that allows that cow to produce a human antibody. And that is going to be produced over and over and over again. So what we get are polyclonal antibodies. Poly, many, cloned, they're all exactly the same. Antibodies, because they all came from this gene that we've inserted. Now, the gene that we inserted is very specific, so I can collect polyclonal antibodies which are specific to, let's say, smallpox. So I can stockpile polyclonal antibodies for smallpox. If smallpox is suddenly within an area of the world, I can use the smallpox antibodies, inject it into the people who are suffering, and that's going to help them to speed up the process. They don't have to use their own immune system. We've got these ready-made antibodies. So human antibodies can be produced in this way. What I should have said is that the cow produces milk, and the human antibodies actually are, they come out within the milk of the cow. So they're quite easy for scientists to extract them from that milk as well. So have a look at this example here. This New Scientist article will take you through that. It's very accessible reading. And again, I'm sure once you've read that, you want to come back and ask me some questions about this. There's one more example here. And this example is really even more cutting edge. It's at the stage now where it's being trialed. And I think it was last year, a baboon had a transplant with a pig heart. Now, there's something called xenotransplantation, very futuristic science, but xenotransplantation uses animal organs to replace organs, for example, in humans. Now, why would you want to do that? Well, there are a lack of organ donors, first of all. So remember the work on the kidney. We said that if your kidneys fail, one thing that you might want to do is have a kidney donated to you to replace the one that's malfunctioning in your body. But that donor kidney needs to be a very good genetic match, because your body will see it coming from someone else, and it will raise an immune response to you. So you're using, in the case of the kidney, a human kidney. That's problematic because you've got a lack of donors in the first place, and then you've also got this idea of the recipient raising an immune response against the donated kidney. So what we're going to do instead is we're going to grow a replacement organ inside an animal which is cloned for that purpose. So what we're going to try and do, and this example at the bottom here talks, it's a Guardian article, and it talks about the story whereby you have a cow, and the cow is generating antigen-free organs, and antigen-free organs means that you're not going to have that same immune response. There's all sorts of laboratories all around the world which are researching this at the moment. It's not obviously common practice yet, but it's coming.